Thank you very much for your introduction. My name is Liu Jing. I will give the presentation on behalf of Dr. Liu Jie. The topic is about the poultry engineered vaccines. The great work is from Bing and his team. The material has been prepared by Bing. I hope that I will try my best to deliver it clearly. Let's begin. There are two parts: technology platforms and innovation application of the novel poultry vaccines. We have 12 technology platforms. The first is reverse genetic, and the second is recombinant protein expression. The third is suspension culture, and the fourth is high density fermentation. I will introduce one by one. We use reverse genetic platform. Establish diverse DNA and RNA variants based on their genomic characteristics and perform process genetic engineering of the viral genome guided by the structure and biologic functions of the key viral proteins. We also construct the H9ARV vaccines strain HRE9. It achieved broad antigenic coverage, non-constitutive mutations. The titer increased 12.9-fold. We do genetic modification of the recombinant DNA. Homology modeling of the tertiary structure was performed for both memory-filled domain of the F proteins and the receptor binding site of the HA protein, followed by the functional side-directed mutagenesis to enhance the replicative capacity of the recombinant vaccines. The general technology we use it to select antigens through step-wise analysis. We systematically select practical antigens that induce dual immune responses, humoral and cellular immunity. Technique 1, gene sequence optimization and expression plasmid engineering through coding pairs optimization and real coding tRNA sublimation. High yield recombinant protein expression was achieved. Technique 2, engineer bacteria strain development. We pioneered novel engineered bacteria strain future integration of cake chaperone gene and knockout endotoxin synthesis gene ICXM, also incorporation of recording system for real tRNA supplementation. For this, enhance the protein translation and folding efficiency while reducing any toxin production. We also use the E. coli platform, and we achieved a high antigen expression and cells with sampling of IV V2G into VLPs. For adenovirus, Fiber 2 was identified as a predominant immune gene through systematic screening. We report the first successful high-yield solution expression of Fiber 2 in E. coli. We also pioneered the development of FAD-based subunit vaccine. A good news is that for the next month, we will launch our first commercial FAD-based subunit vaccine. It will be the first commercial adenovirus subunit vaccines in the world. We also use the equalized platform to establish the first EDI subunit vaccine. The same platform we developed the first IC subunit vaccine. Technique 2, engineering of bacteriovirus expression vectors. By simultaneously knocking out the competition expression gene P10 and the protein secretion inhibiting gene GA and the YCAS, also the integration site of genetic stability empowering gene FP25K and DA26. We engineer the stable and high efficiency bacterial virus vectors. This optimization results in more than fourfold enhancements in gene transcription efficiency and protein secretion capability. For the bacteriovirus platform, we do expression of the ARV-H9 antigen protein. It achieved the co-expression of three immunogenic genes, HA, HNA, and M1. Also, we have do the expression of ARV-H5, H7 antigen protein. We have established the H5 re11, re12, and H7 re2 subtypes. For the recombinant protein expression platform, we have CHO cells platform. Optimize the selection genes and hybrid promoters compatibility with the host cell system enable high efficiency, high level expression of the recombinant proteins. Use CHO cell, we do expression of the IBV antigen inclusion. We also established the ILTV antigens. We high expression stable cell lines can produce GD and GD-GB fusion proteins. It can provide complete protection in avian models. The third platform is the suspension culture. The core technology one is cell sorting. Core technology two is culture process optimization. A control aeration strategy integrated with some continuous. The part two, innovation and application of novel poultry vaccines. A strategy development of novel poultry vaccines is we plan 5 series: ARV multi-trivalent vaccine, IBDV multi-trivalent vaccine, and NV genotype 7 multi-trivalent vaccine, and ADV-EDS multi-trivalent vaccines. Also, the poultry bacteria disease multi-trivalent vaccines. For the ARV-H9 renal subunit vaccine, you can see that serum antibody indicators is equal or more than 9 log 2. It can effectively prevent the viral shedding post the challenges. Also, the immunity developed by 40 days post the vaccination with duration exceeding 6 months. The IBD VP2 protein form. subunit vaccine. It can provide 100% protection efficiency against the IBD health challenge, and also immunity develops by 14 days, and the duration can exceed 8 months. NDV genotype 2, genotype 7. For adenovirus, we already have 3 adenovirus-related unique vaccine registration certificate, 2 class 1 in novel veterinary drugs. For adenovirus, immunity developed by 14 days post-vaccination, and the duration exceeding 12 months. Immunity developed by 14 days after T5 protein vaccination. No adverse effects on the egg production rate and quality post-challenge. You can see that immunity develops by 14 days post-vaccination. The duration exceeds 14 days. Only one single vaccination provides protection through entire life cycle. IC subunit vaccine. Compared with the whole bacterial vaccines, it has lower endotoxin levels and superior safety. IC subunit vaccine demonstrates superior protection in early life chicks compared with the commercial vaccines. The MSS vaccine achieved 17 lesion reduction rate post-MS challenge. The IBV as protein immunization effectively prevents virus-induced cellular damage. The live week uses active triggers cellular movements in vaccinated chickens at 5 days post-infection, and the right is the cellular loss in control chickens at 5 days post-infection. For the IoT subunit vaccine, CH expression system after performance equalize system. 21 days post-vaccination, a non-perfection chickens seropositive for the antibodies, and 28 days post-vaccination, perfection chickens seropositive for antibodies. Post-challenge, zero chicken exhibited clinical disease. Thank you for your attention.